Hello and welcome to the fourth longevity snapshot, where we will take a look at a very comprehensive study on life-extending molecules in worms and the variables which can affect their results, leading to some profound implications. My name is Chris Linnell, and I'm the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do, then please check out the links below this video, where you'll also find a link to the study being reviewed here. Also, please give us a like and subscribe to our channel. Every week, I scour PubMed for the latest longevity studies, and hardly a week goes by when I don't see a study claiming that a certain molecule extends the lifespan of the small roundworm C. elegans, which is one of the most basic animal models in longevity research. But I always ask myself, how relevant and accurate are these results? And truth be told, I don't give them much credence until they've been replicated, at least in mice. The following study caught my eye because it seems to be the most comprehensive study looking at all the variables going into C. elegans longevity research that I've seen so far. And it throws up some intriguing questions for longevity research in general. In this study, researchers evaluated 16 molecules well documented for extending C. elegans lifespan. They tested them under various conditions, as well as combining some of them to see if there were any synergies, tested some of the best performing ones in the fruit fly to see if there was any cross-species applicability, and also did a screen where they identified some new life-extending molecules in C. elegans. So let's take a look at the first figure. Here you can see the 16 molecules tested, from aspirin in the top left all the way down to urolithin A in the bottom right. And in each of these survival curves, the black line is the control and the blue line is with treatment. On the right-hand side, you can see the median lifespan results of those 16 molecules under different concentrations. The first variable which they tested was the food given to C. elegans. C. elegans feeds on bacteria, and the strain of bacteria is called OP50. And in this case, they were given live OP50 bacteria to munch on. Now, you should see that while these 16 molecules were all documented to increase the lifespan of C. elegans, not all of them did so in this test. So, for example, aspirin, the two lines are basically the same with glycine, and perhaps surprisingly, the same with rapamycin. Now, I won't dwell on this first figure because the results are shown again in the next one. The second variable which they tested was, instead of giving C. elegans live bacteria, they gave them dead bacteria to eat. And you can see the survival curves of the same 16 molecules in B, with the median lifespan results under varying concentrations in D. In C, and I'll make it bigger so we can see better, you can see the median lifespan increase between the live and UV-killed bacteria food. For some, such as aspirin, there was no increase in lifespan either for the live or dead bacteria food. Some, however, saw an increase in lifespan when given live bacteria, but that increase disappeared when they were given dead bacteria, such as captopril here, which is an ACE inhibitor. The reverse was also seen, where there was only an increase in lifespan when the animals were fed dead bacteria, as simvastatin shows here. For 6 of them, there was an increase in lifespan under both conditions. These were caffeine, doxycycline, GSK 2126458, which I'll call GSK from now on, LY 294002, which I'll call LY from now on, metformin, and resveratrol. As you should also notice, more of the molecules showed a lifespan increase when the worms were fed live bacteria than dead bacteria. One reason for that could be is because some of the molecules might protect C. elegans against the bacteria, because other studies have shown that C. elegans lives longer when given dead bacteria compared to live bacteria. Not only is there a difference in lifespan depending on the food source, but also in the solvent used to dissolve all of these molecules, which you can see in A on the left here. DMSO is the standard solvent used, and on its own, it can also increase C. elegans lifespan, as you can see comparing the blue line with DMSO compared to the control line here. And also the concentration used has an effect. As you can see on the left, it's 0.1%, and on the right, it's 0.5%, and the 0.5% concentration increases the lifespan more. Now, above 5%, it actually kills the worms. The researchers also looked at what effect these 16 molecules had on the bacteria used for food. And out of the 16 molecules, only resveratrol, LY, and urolithin A had no effect on the bacteria, adding yet another wrinkle to these studies. Next, they took 4 of the best performing molecules and tested them in pairwise combinations, once again using live and dead bacteria as the food source. You can see the lifespan curves at the top here, but the results are clearer when looking at the graphs at the bottom. For a couple of them, there was a synergistic increase. So for example, doxycycline and GSK and LY and GSK showed a considerable increase, as you can see by this blue line compared to the black lines, which are those molecules on their own. Those two results are for live bacteria, and for the dead bacteria, only resveratrol and doxycycline showed a synergistic increase. Others didn't show a synergistic increase, and in fact, for 2 of them, resveratrol and GSK and resveratrol and LY, in fact, showed a marked decrease compared to the molecules on their own. And in fact, resveratrol and GSK showed a reduction in lifespan. Next, they tested 2 of the molecules, LY and resveratrol, in 3 other organisms. One, Ju775, which was a different strain of C. elegans, and also C. tropicalis and C. briggsea, once again with live and dead bacteria. So you can see the survival curves on the left-hand side, and here on the right shows the median lifespan. And they had differing results dependent on the organism and the type of bacteria used. So both LY and resveratrol increased the lifespan of Ju775 with live bacteria. However, resveratrol didn't increase it under the dead bacteria. Tropicalis only had its lifespan increase with the dead bacteria. And C. briggsea had an increase with live bacteria. But when they were fed the dead bacteria, they supposedly tried to climb out of the Petri dishes. They didn't like the taste, I guess. That's why there's no graph here on the bottom right. In C here, they also tested LY against 2 mutant worm strains, AGE1 and DAF2, and AGE1 had the gene P13K knocked out, and DAF2 has the gene IGF1 knocked out, and you can see there are long-lived strains of C. elegans, but in the conditions tested, LY only increased the lifespan of AGE1 worms fed live bacteria. You can see here in this survival curve. Next, they looked at giving 7 of the molecules to C. elegans at different stages in its life cycle. According to the authors, and I didn't know this before, most drugs are given to C. elegans starting at L4, which I believe is the 4th larval stage. And in this experiment, they gave C. elegans the molecules 1 day after they became adults, once again with live and dead bacteria, as you can see in the survival curves at the top. Looking at the median lifespan between L4 and day 1, for the live bacteria, doxycycline showed a comparable increase in lifespan between L4 and day 1, whereas the others showed a decrease. For the dead bacteria, once again, doxycycline showed a comparable increase, as did caffeine and GSK. The other 3, once again, showed a decrease. Next, they tested 5 of the best performing molecules also in fruit flies. And nice to see that they also used both male and female fruit flies to see if there was any sex differences. And you can see the survival curves at the top. The median lifespan increase under varying concentrations in B, and we'll take a closer look at the results in C here. So as you can see, doxycycline showed a comparable increase in lifespan across species, across the sexes of the fruit flies, and across the 2 different food sources. Metformin also showed a comparable improvement. However, LY didn't show any increase in female flies. Monocycline only showed an increase in live-fed C. elegans, and whereas rifampicin only showed an increase in live-fed C. elegans, it also showed an increase in male and female fly lifespan. Lastly, they did a screen of hundreds of molecules to see if they could find any life-extending ones among them. And you can see the results here at the top with the live bacteria-fed ones and the UV-killed ones on the right. And the results showed either increases or decreases in lifespan. Now, out of the live bacteria-fed C. elegans, only 34 out of the 470 molecules tested showed a lifespan increase. And in the dead bacteria-fed C. elegans, Only 39 out of the 592 molecules tested showed an increase in lifespan. And that was either 7.2% or 6.7% out of all molecules tested. Then they tried to replicate those positive results numerous times and came up with 11 molecules showing robust extension for the live-fed C. elegans and 6 molecules for the dead bacteria-fed C. elegans. And out of those, 5 molecules increased lifespan under both conditions. Looking more closely at C at the bottom here, those 5 molecules were the antifungal mecanizole with a 26% increase in the live fed and a 23% increase in the dead fed. The antiparasitic fenbendazole with an 8% increase and 57% increase. The PDE5 inhibitor tadalafil, better known as Cialis, showed a 28 and 26% increase. The MAO-B inhibitor used in Parkinson's, safinamide, showed a 36 and 14% increase. And lastly, the tyrosine kinase inhibitor AG1478 showed a 14 and 17% increase. Now, out of those 5 molecules, only AG1478 had been characterized as being a lifespan increasing molecule. So the other 4 are new molecules in that regard. So what does this all mean, and why did I think this had profound implications? Well, firstly, it shows that you can't trust single studies in C. elegans on their own. This study showed that there are so many variables which can influence C. elegans' lifespan. The concentration of the solvent, whether or not their bacterial food source is dead or alive, the influence the molecules have on the bacteria themselves, at what stage in C. elegans' life cycle the molecules are administered, the concentration of the molecules, not to mention that different species of roundworms have different results, and that the results might not cross the species barrier. It has certainly made me much more wary of trusting any C. elegans lifespan results. And I hope that Matt Kaberlein and his million molecule challenge, where they're using the worm bot to try to screen as many molecules as possible in C. elegans, has taken note of this paper. I'm sure he has. Finally, and this pertains to us, be very careful when trying to combine supplements. As we've seen, molecules which on their own increase lifespan don't necessarily do so when combined. In fact, most of the combinations did not lead to an increase in lifespan. Indeed, some of them actually led to a decrease. The ones which synergized effectively were few and far between. So please be careful out there. And that marks the end of another longevity snapshot. I'd love to hear your thoughts on whether or not you agree with me that this was a profound study, so please put them in the comments below this video. All that remains is for me to ask you to please like this video and subscribe to our channel. So until next time, goodbye.